Hi everyone. I'm going to talk to you about how we are using geospatial analytics and machine learning methods for improving pest survey design. My name is Kumar, and I am a quantitative risk analyst at the Phytosanitary Advanced Analytics Team, or PAAT, at APHIS Plant Protection and Quarantine, PPQ, in Raleigh, North Carolina. First, I would like to thank my colleagues, Dr. Derek Waller and Dr. Larry Yak from PPQ Science and Technology Phoenix Lab in Arizona for providing me wonderful grasshopper photos that you would see throughout my presentation and helping me with grasshopper biology. Now I'm going to talk to you about a case study of forecasting grasshopper populations in the Western United States, which can potentially help us in strengthening PPQ domestic survey programs. As some of you may know, APHIS PPQ surveys the entire country every year for hundreds of emerging pests and diseases, and we have some preliminary evidence that we can significantly improve the detection accuracy of our surveys by incorporating large-scale geospatial analytics and machine learning methods. The primary goal of this project is to improve domestic pest and disease survey programs, which will lead to more efficient and better safeguarding of American agriculture. Now you may be wondering why we need grasshopper forecasting tools. Well, have you ever taken a walk in the grasslands in western US in July and August? I did when I lived in Fort Collins, Colorado, and so do many of my colleagues in the western US every year monitoring grasshopper populations. So when you walk across the grasslands, you would see many grasshoppers hopping around in front of you, as you see in this recent video from my, that was shared by my colleagues from Montana. So there are approximately 400 grasshopper species in the 17 western US states. However, most of them are harmless, but a small percentage are considered pest species that can cause significant damage. And a large number of grasshopper species can eat tons of grass per day and cause millions of dollars in economic damages. Now, grasshoppers can damage rangeland habitats. They can reduce forage for cattle and other grazing livestock and affect wildlife and rangeland health. For example, look at these two rangeland photos taken by my colleagues in Phoenix Lab in the summer of 2014. They are both a few miles apart in New Mexico and receive similar levels of rainfall. However, the top photo shows damaged grassland from grasshopper feeding, and the bottom photo shows no damage. We see a significant difference in these two photos. Top photo shows reduced grass biomass compared to the bottom photo. Grasshoppers can also devastate cultivated crops such as alfalfa, wheat, barley, and corn. The year-to-year changes in weather conditions can result in grasshopper outbreaks. The outbreaks in an area is defined based on the number of grasshoppers per square yard. In this case, we define if you find 15 grasshoppers or more per square yard as outbreaks. The outbreaks occur when the environmental conditions are good for grasshoppers. For example, warm and dry spring, as it happened in Montana this year. When an outbreak occurs, it can have damaging effects on rangeland health. Therefore, PPQ and our many partners have been working on developing novel and improved methods of grasshopper detection and management. Each year, APHIS provides approximately $5 million to support rangeland grasshopper and Mormon cricket suppression program. However, as we know, the climatic conditions fluctuate from year to year. So in some years, we find more grasshoppers than normal. Therefore, we need more funds to control outbreaks. For example, this year USDA provided an additional $2.8 million in June to control outbreaks. Overall, APHIS's grasshopper and Mormon cricket program helps to minimize damage and protects agricultural resources that's worth approximately $8.7 billion. So for this case study, we are using a very mature and data-rich program called Grasshoppers and Mormon crickets. And then we will generalize our approach to all applicable pest and disease surveys. This massive data set is a result of over many years of hard work by many of my colleagues from all over 17 so-called grasshopper states. And in this presentation, I will show you how geospatial analytics and machine learning methods can help us forecast grasshopper populations more accurately to identify population hotspots, which can then be treated more rapidly, thereby reducing the potential for outbreaks and safeguard rangeland habitats. So APHIS PPQ conducts surveys every late summer or fall across 17 western US states. Data from these surveys are used for generating a grasshopper hazard map, which is used by APHIS and land managers and owners to assess whether treatments may be needed. APHIS treatments are only conducted when directly requested and funding is available. The current grasshopper hazard map that you see on upper right corner of this slide is based on previous year's grasshopper survey data, and it generated in ArcGIS using kriging, which is an interpolation method to generate information beyond data sites. The premise behind this map is that previous year's grasshopper adult population would lay eggs in the fall that would hatch in spring and contribute to next year's grasshopper population. However, this map does not consider any climatic, landscape, or other biological factors. Therefore, we are exploring science-based advanced analytical tools such as geospatial analytics and machine learning methods to improve this map to enhance decision making. In this ongoing project, we have 5 major objectives, and our first objective is to develop grasshopper population forecasting model for 4th north central counties in Wyoming, which are shaded yellow in this slide. Through this pilot project, we wanted to demonstrate and use the use of APHIS grasshopper data to develop biologically informed grasshopper density spatial predictive model. And we wanted to test whether we can make forecast. That means make future projections of grasshopper density in these 4 counties. We selected these 4 counties because of data availability and processing time, and I'll show you the results from this objective. Our second objective in this project is to develop grasshopper outbreak predictive model for the entire western United States. And I have acquired historic grasshopper survey data from 2002 to 2019 that my PPQ colleagues have collected and archived in a geodatabase. And we are collecting a time series of climatic and other environmental data layers for variables that can potentially influence grasshopper outbreaks. I will then use a combination of statistical and machine learning methods for predicting the probability of grasshopper outbreaks in the western US. Our third objective is to develop potential distribution models for 12 economically damaging and widespread rangeland grasshopper species in the western US. For this, we are using a maximum entropy based machine learning model called Maxent. For an example, you see here a map for one of the Pest grasshopper species in Wyoming, which was generated using Maxent. We want to do this to identify and map areas where these 12 most damaging species can potentially occur, because these species often cause outbreaks in the western US. And as we know, different species respond to climatic conditions differently and have slightly different life histories. Therefore, a species-specific approach might provide us additional insights. Our fourth objective is to develop grasshopper population forecasting models for different eco-climatic zones in the western US. As we know, there are regional climatic and topographic differences in these 17 western US states. So a forecasting model developed for northern plains may not work for the central plains or southern plains or vice versa. Therefore, I'm using neon domains, which are shown here in red polygons, to develop 8 regional grasshopper forecasting models. Our fifth and the most important objective is to extend this forecasting approach to other applicable PPQ pest and disease surveys. This slide here summarizes a general predictive spatial modeling framework that I'm using throughout this project. First, we have the data from field surveys on species occurrences or abundance with GPS coordinates. Next, we bring this data into an Excel spreadsheet, as you see here, and a GIS software. And then we generate GIS data layers for different environmental variables that can potentially affect species distribution and abundance, such as climatic and topographic factors. We then combine these two using a statistical or machine learning algorithm, and the result of this exercise is a predictive map showing the predicted abundance or probability of presence for a species in your study area. Okay, so for those of you who are not familiar with machine learning, you may be wondering, do machines really learn? Because we're calling it machine learning. Well, the short answer is yes. Machines learn by studying observed data to detect patterns. We can train a machine learning algorithm using observed data and then make predictions or forecasts to new data or scenario. And that is what exactly I'm doing using two machine learning methods in my project here. I'm using the booster regression trees method for modeling grasshopper density and outbreaks, and I'm using machine learning or Maxent model um, for modeling potential distribution of individual grasshopper species. Now I'll talk to you briefly about the methods we use in Objective 1. We obtained grasshopper abundance data from 56 common data sites in 4th North Central counties in Wyoming. Here is an enlarged map of those counties with black dots representing those common data sites. We selected a number of static and dynamic environmental variables that can potentially influence grasshopper density. The static variables are the ones that don't change over time, such as soil types, slope, aspect, and elevation, as you see on the left here. And the dynamic variables are the ones that change over time, such as temperature, precipitation, and soil moisture. We selected these variables based on grasshopper biology, such as the timing of egg laying, overwintering, and egg hatching. This is a long list of those static variables that we considered in our study, including topographic, land use, land cover types, and soil characteristics. We considered three sets of dynamic environmental variables in our modeling. First set included satellite-based vegetation indices from NASA. Second set had monthly climatic variables from PRISM climate dataset. And the third set included soil moisture data from NASA, and that is hosted by Esri in their Living Atlas. Here is a long list of those dynamic variables. As you can see, we had many variables, almost 70 plus variables, and 6 years of grasshopper density data. This prompted me to explore the machine learning modeling that can handle large and complex data sets. Well, since it's difficult to explain all the methodological details in this short presentation, I will directly go to the results from our first objective. But if you have any questions, please feel free to contact me after the presentation. I know this seems like a busy slide, but let me walk you through it. The top two figures, A and B, show the observed grasshopper density represented by varying size of black circles. And the bottom figures, C and D, show predicted density using general linear model or multiple regression model. The model was calibrated using 5 years of grasshopper density data from 2012 to 2016, and then projected to 2017 to make forecasts. Both models, as you can see, were highly statistically significant, and goodness of fit index R-squared value was 32% for the calibrated model and 8% for the forecasted model. However, when I used machine learning model called booster regression trees, the R-squared value is almost double for both models. And this model also had higher granularity, as you see here, providing us more details in model predictions, as you can see in these figures C and D. Overall, we showed that it's possible to make grasshopper density predictions in space and time. That means we can make forecasts of where and how many grasshoppers you might expect in your study area next year. So to summarize, using geospatial analytics and machine learning methods, we have demonstrated that there is a better way to do surveys that improve our likelihood of detecting grasshoppers populations, so that control treatments may be optimized. We expect that we will be able to improve grasshopper density forecasts in all grasshopper states in the Western US and provide the basis for improved surveys for all PPQ domestic programs in the future. The results from this study will be published in PPQ internal report that would be available upon request. We are also preparing a couple manuscripts that we plan to publish in peer-reviewed journals. I have many people to thank. As you can see the busy slide, a lot of people who are pretty dedicated and hardworking have contributed to this work. And first of all, I would like to thank my supervisors for their continued support and giving me an opportunity to present, to work on this project, and many people who have spent many years working on grasshopper environment crickets. Thank you for your attention. I would be happy to take any questions.